I will close the door. Good morning. Shall we start? Thank you. Dear colleagues, I'm very pleased to welcome you to Belgrade, to University of Belgrade and the Faculty of Philosophy, which is hosting this year's conference. Organizing such a large and important conference is a great undertaking, especially in the specific and very sensitive socio-political circumstances that currently exist in Serbia. Thank you all for coming to the conference, because we also see it as a support to our educational and research community, and we need that support, so thanks once again. Now, I am pleased to announce our plenary speaker, colleague Dr. Ben Williamson. Dr. Williamson is a senior lecturer and co-director of the Center for Research in Digital Education at the Morris House School of Education and Sport, University of Edinburgh. He's the author of Big Data in Education, the Digital Future of Learning, Policy and Practice, and editor of the World Yearbook of Education 2024, Digitalization of Education in the Era of Algorithm, Automation and Artificial Intelligence, and also co-editor of the journal Learning Media and Technology. Ben has 40-45 minutes for his presentation, and after that we will have, I suppose, 10 to 15 minutes for comments, reactions, discussions. Ben, the floor is yours.
Thank you, Anna. Thanks, ECER, for having me. It's a pleasure to be here. I thought I'd start with a story. Earlier this summer, a startup technology company from Silicon Valley announced the launch of a genetic IQ test for embryos. So Nucleus Genomics, which you can see here, claimed that its pre-implantation embryo testing platform was, and I quote, the first genetic optimization software that lets parents see and understand a complete genetic profile to select an embryo. Now, the intelligence testing part of the platform, amongst the other medical diagnostics, implies then that prospective parents could select an embryo with high predicted IQ and the best possible chances in school. That is, of course, if you're a wealthy parent with all the dollars you'd need to select a super baby. Now, we could, of course, just respond to this and say it's modern eugenics, that it's just snake oil, and other critics have already said that. This is clearly about privileging or promising the most privileged that they can improve their own gene pools while the company profits from scientifically misleading marketing. But my reason for starting with this example is because there's been a dramatic increase in interest in the last 10 to 15 years in how genetic data of this kind might be used in education. So as you can see from the images here, there's now ongoing discussion about the potential of integrating genetic data technologies and methods into education practice and policy. And one reason for this interest is because new genomic technologies like consumer DNA testing platforms, like the one from Nucleus, could be used to collect masses of genetic data about children, it is claimed. So one expert advisor to Nucleus is a well-known behavior geneticist who's written an entire book about the idea of studying children's DNA from consumer genetic testing. And several similar books, and now many, many academic papers, are kind of convening on one key idea. And that idea is that genetic data, collected and analysed with cutting-edge technologies, could transform our understanding of learning and educational outcomes, and could also be used to design genetically informed education systems and practices. So this is what I and other colleagues have begun characterizing as the geneticization of education, or genomically informed education, or as I prefer to term it, educational genomics. So educational genomics, as scientists have claimed, aims to uncover, and I quote here, the underlying genetic architecture of academic achievement and advance a more comprehensive, biologically oriented model of individual differences in cognitive ability and learning. So together with educational neuroscience, educational genomics represents what I'm calling today the emergence of the bio-edu-data-sciences. And that is highly data-intensive biological sciences of educationally relevant behaviors, processes, and outcomes. The bio-edu-data-sciences attempt to look into the body, into brains and DNA, using advanced genomic and neurotechnologies to data mine neural and genomic bioinformation. And they are highly consequential, as well as controversial, for educational research, for policy, and for practice. So I'm going to spend my time here today outlining some of those consequences and controversies associated with this kind of biologization of education. I'm going to focus on educational genomics in the interest of time, though if you want to read a bit more about our work on educational neuroscience, we have a couple of papers online. First, a big thank you to the Leverhulme Trust, who have funded our project on the birth of the Bio-edu-data-sciences, and also a huge thank you to my colleagues on the project, and they are the political sociologist Dimitra Katuza, the social and political geographer Jessica Pikett, and the sociologist of biomedicine Martin Pickersgill. None of them are here today, but I really, really couldn't be doing this talk without their collaboration over the last 4 years. So, conceptually, we come from a more social scientific, sociological perspective, and we're grounded in social scientific studies of the biological sciences. A collection of core readings are right here. So biology, and especially human genetic sciences, have undergone a significant transformation in the couple of decades since the human genome was sequenced. So researchers, like the ones you can see here, refer now to a post-genomic condition after the sequencing of the genome, where wet, biological, embodied substances and materials are no longer the main objects of biological investigation. Instead, biologists increasingly deal with dry, digital bioinformation that's stored in databases and studied using bioinformatics software in computer labs. So as Hallam Stevens phrases it, this computerization of biology has changed what biologists do, how they work, what objects they deal with, and, perhaps most importantly, what kind of knowledge they produce. So as biology has become increasingly data-centric, as Sabina Leonelli argues, biological knowledge production has been reshaped by bioscientific technologies, databases, and algorithms. How biologists think about biological objects has changed as they've become digital biological objects of attention. Now, these bioinformational ways of doing and knowing biology have also been adopted into other fields, including psychological and social scientific methods and practices, particularly in the form of behavioral genetics and social science genomics. Now, very quickly, Behavioral genetics is a branch of psychology that traces complex human behaviors and traits back to their kind of underlying genetic base. And social science genomics, or sociogenomics as some call it, tries to understand the genetic influences on social and economic outcomes, as it were, tracking the pathways from cells to society. Now, both behavioral and social genomics are concentrating significant attention on education. They've adopted the genomic, data-centric style of bioinformatic investigation to discover the genetic basis of educational outcomes, achievements, and cognitive ability and intelligence. So this is a big shift, I think, in how research about educational matters is done and by whom it is done, what data are collected, what methods are used, and what kind of biological knowledge is now being produced about those educational matters. So our overall conceptual orientation is to see educational genomics as an inventive science. That is, in our argument, it is inventing a bioinformational account of learning and education. So educational genomics does not just make the kind of biological substrates of learning outcomes visible at the molecular scale. What it does is it generates, fabricates, or invents them through the use of bioinformation or biodata. And it then promotes these bioinformational conceptualizations of learning or educational outcomes as the basis for policy and practice in schools. So its objective, we might say, is to make saliva samples from DNA swabs the basis for educational policy and classroom interventions. Okay, so the rest of the talk is now going to be framed around what I'm referring to as the 5 I's of educational genomics. And those 5 I's of educational genomics are infrastructuring, instrumenting, informating, intervening, and inventing, with a sneaky half-6th I at the end. So, to start with infrastructuring. This refers to the ongoing construction and maintenance of a system of scientific investigation and knowledge production. So basically, you can't get from the spit samples in the tube to social policy without a vast infrastructure for collecting data and analysing it and presenting it. So what we're doing here is drawing on standard kind of science and technology studies accounts of knowledge infrastructures, which see them as relational systems of people, organisations, practices, and technologies that underpin how scientific knowledge and scientific knowledge claims get produced. And this kind of infrastructuring also highlights then how objects of scientific investigation are bound up with computer and information systems, which themselves mediate and shape how particular objects are conceived or interpreted and explained. So, in our view, educational genomics can only exist because a knowledge infrastructure for carrying out investigations and reporting findings has been constructed over the past 15 years. So educational genomics is not a discipline, it's not a field, it is made up of a wide range of individuals, networks and associations across other disciplines and fields. But it is the infrastructure, the knowledge infrastructure, that holds them together and sustains their scientific investigations. And central to this infrastructuring of educational genomics, then, are databases of biological information, or bioinformation. And databases perform what we have called a harmonizing function in educational genomics. And we mean that in 2 ways. Firstly, the collection of vast databases for analysis has involved the harmonization of a range of diverse data sets. So this kind of data harmonization function is a key aim of this organization, the Social Science Genetic Association Consortium. The SSGAC It's the largest international network of scientists involved in social and behavioral genomics research, and it has produced the largest sample studies of the genetic basis of educational outcomes, with the most recent sample being 3 million people. Now, the major sources for these studies are the UK Biobank, which is a publicly funded data bank about genetic information from a British population sample, and the other is 23andMe, the Silicon Valley consumer genetics company that has the planet's largest private biobank, and there are other smaller cohort data sets included in the SSGAC studies too. So a lot of harmonization is involved in making these data sets compatible for research. And that includes a whole bunch of legal, financial, logistical, and technical practices and personnel. Databases, though, also facilitate the formation of what we've termed after Ackerman and colleagues, harmonious scientific collectives who have shared aims, methods, theories, ways of seeing, and ways of working. So as it itself says, the SSGAC is a platform for interdisciplinary collaboration and cross-fertilization of ideas, as well as a repository of harmonized data. So in this sense, the knowledge infrastructure of educational genomics has been harmonized by databases. The scientists that enact educational genomics have been brought into harmonious relations in the form of these interdisciplinary associations and networks that operate internationally and at scale to analyze very large data sets of bioinformation. Now, studying all of that bioinfo from those databases requires a whole bunch of scientific instruments. And these instruments in this space are largely described as bioinformatics, which refers to the synthesis of biological with statistical, computer, and data sciences and their applications. From our perspective, the scientific instruments used here, though, are not simply, as it were, tools that help scientists do their analyses. Instead, scientific instruments actively mediate how reality becomes present to and is treated by scientists, as Baz de Boer and colleagues put it. So the bioinformatic instruments used in contemporary biological research, the quantities of data processed by computers, the algorithms needed to deal with them, make a qualitatively different kind of knowledge. And that has significant implications in terms of how biological phenomena are seen and understood. So scientific instruments, to put it bluntly, co-produce genomic facts while excluding other ways of seeing and understanding biology. Now, the bioinformatic instruments that are commonly used in educational genomics include microarray chips. These are little glass slides that are used to identify an individual's genetic differences from a wider population. And what microarrays are said to surface are, wait for it, single nucleotide polymorphisms, SNPs, or as we'll refer to them, SNPs. These SNPs are tiny molecular differences in DNA that, it is claimed, interact to influence complex behaviors, traits, or outcomes. Now, the invention of these microarray instruments has enabled researchers doing educational genomics to claim that, here's another quote, molecular genetic research, particularly recent cutting-edge advances in DNA-based methods, has furthered our knowledge and understanding of cognitive ability, academic performance, and their association. Now, the 3 million sample study by the SSGAC claims to have identified the genetic architecture of educational attainment from such microarrayed data. Now, this genetic architecture consists of statistical associations between around 4,000 interacting SNPs. So it's this claim that we now know the molecular genetic architecture of learning outcomes. That is why, basically, those doing educational genomics find microarrays so compelling. So compelling that some scientists have even called them learning chips and characterized microarrays as DNA fortune tellers that could be used to predict educational outcomes in genetically sensitive school systems. But the biological realism of such claims becomes perhaps a bit more questionable if we recognize that these so-called learning chips are products of commercial biotech companies that have changed how biologists understand genetic variation. So as Ramya Rajagopalan and Joan Fujimura put it, the design, statistical power, and technical constraints of the chips have shaped the definition and significance of human genetic variation. Microarrays, they say, have locked in the primary conceptual frameworks that humans should use to consider, organize, work with, and ultimately act on genetic differences. The association of molecular genetic variation with educational outcomes has therefore been locked in to educational genomics by the design and the constraint of these DNA microarray chips and the kind of data analyses that they enable. So in this sense, instrumenting educational genomics with microarrays as well as other bioinformatic instruments doesn't necessarily signify, as is claimed, a more accurate, biologically realistic understanding of genetic differences and how they predict educational outcomes. The design of the instruments, to a certain extent, produces those differences, making them visible as bioinformational artifacts that stand as statistical proxies of biological mechanisms. Now, the third of my 5 I's of educational genomics then is informatting. And it means, again, 2 related things. Firstly, the transformation of biology into information. And second, the specific ways that this bioinformation is formatted in standardized form. And Colin Koopman's work on genetic data formatting is key here. So as Koopman argues, one thing the genetic sciences do with tremendous success is to format us as subjects of genetic data. In being formatted, rendered, or organized as genetic data, we come to be defined by that data. Others can address us, deal with us, or even impose burdens and benefits on us in terms of the genetic data we've become. And then Koopman argues that data are formatted in 3 stages: as input data, as methods or formats for data storage, and as formats of data sharing and presentation. So how data are formatted at all 3 stages is consequential for how individuals and collectives are known genetically and then treated as a result. So the specific data format centered by educational genomics is called a polygenic score. So a polygenic score is basically the sum total of all the thousands of differences captured from one's microarray data. So then, the microarray data are formatted by the design and constraints of the chip at the point of data input. Polygenic scores are then produced through specific methods that are enacted by particular bioinformatics instruments which render the data into a particular format. And that format is a single numerical score for the educational outcome or trait of interest. And that formatted score can then circulate as a seemingly genomic fact of one's predicted educational achievements. Okay, so now in educational genomics, the most common polygenic scores are produced for educational attainment, and that is described in one of the most influential papers as a phenotype affected by thousands of genetic variants, each responsible for a minuscule fraction of individual differences. And the graphic here is from one of the SSGAC's large studies of educational attainment. And it produced polygenic scores for over 1 million people in this study. And the graphic depicts just some, of the thousands of genetic locations in the brain that are said to be associated with differences in educational attainment. So the task of the scientist, putting it simply, is to add all of these up into a polygenic score that is then said to be an authentic genetic signal of one's future educational attainment. So that's a really computationally intensive process of decoding complex genetic data into the simplified format of the score. And the SSGAC's work is particularly significant here because the polygenic scores from its studies are now basically the reference models that are used in a whole range of other educational genomics studies. So we've turned this informating, because it's a highly informationalized process that basically squeezes the biological correlates of learning into the simple format of the polygenic score. And then how data are informated makes a considerable difference to how it can be presented, interpreted, and how ultimately it might be used for intervening in educational policy or practice. So we come to the fourth I, which is intervening. And in the paper that we're working on, finishing just at the moment, we've identified, we think, 4, maybe 5 ways that polygenic scores have been proposed for use in either education policy or practice interventions. These 2 press articles, I think, are indicative of considerable press interest as well as political interest, or some political interest, in the application of education, the potential application of genetics knowledge. So I'm going to run through 4 of these proposed forms of intervention based on the use of polygenic scores. So the first is what we're calling, or the first way that polygenic scores have been proposed for use in education is through this idea of precision education. And that refers to a kind of customization of teaching on the basis of a student's personal polygenic score for educational achievement. So, as the authors of a recent paper have put it, the DNA revolution has made it possible to predict individual differences in educational achievement from DNA, and their use has been proposed for predictive enrichment. That is, identifying which populations are most likely to benefit from a given treatment. So on this basis, it's been claimed that polygenic scores are key for personalized or precision learning as they predict pupils' profiles of strengths and weaknesses, which offers the possibility to intervene early to prevent problems and promote promise. But this, I suppose, it's supposed to be a kind of seductive vision of pinpointing children for precisely targeted pedagogic support. It also requires a certain kind of hierarchical arrangement based on measured genetic differences, which perhaps this graphic at the bottom helps illustrate. So as Callie Burt has phrased it, polygenic scores are actually rankings. They're positions on a scale of genetic associations with socially valued outcomes. So polygenic scores used in this way for precision learning then are then a technology of ranking, or we might call it ordinalization. You can plot students onto a genetic hierarchy. So precision education represents what we've called the bioinformatic ordinalization of students by polygenic scoring. And this means ranking them on an ordinal scale of genetically predicted outcomes and then targeting them for intervention. The second proposed use of polygenic scores is as screening tools for group differentiation in schools. And this is most clearly put in a 2021 report for the Early Intervention Foundation in the UK, which claimed that it's increasingly possible to identify at birth children who have an elevated likelihood of outcomes such as struggling at school or being diagnosed with a learning, behaviour or mental health condition. So, as the authors of a related research study elaborated, polygenic scores then could be used as an additional tool for universal screening systems in authentic school settings alongside the progress monitoring tools that are already regularly used in schools to differentiate based on group needs. So this form of polygenic screening emphasizes then the potential to group children into distinct genetic clusters identified by a kind of shared distribution of SNPs. And we conceive of these groupings, as they're conceived here, as the designation of polygenic biosocialities. So this concept of biosociality, as some might know, refers, as Ian Hacking has phrased it, to how an avalanche of genetic information available about individuals and populations has enabled the identification of groups according to genetic indicators associated with a biological condition. Or, as Paul Rabinow has argued, through the use of computers, individuals sharing certain traits or sets of traits can be grouped together through a technocratic administration of differences. So polygenic scoring here means the production of polygenic biosocial groups that are classified, sorted, and differentiated by genomic screening technologies. A third proposal for using polygenic scores is for cost savings and efficiencies in education systems. So polygenic scoring here would operate as a kind of biostatistical indicators of whether a policy is cost effective and for whom, how, or why a policy works in a particular way. So in the book, The Genetic Lottery, KP Harden proposes using genetic data to accelerate the search for effective cost-saving interventions in schools. And she argues, quite provocatively, that interventions in schools that are usually based on social sciences alone waste time and money by failing to even consider the role that genetics plays in how children's lives turn out differently. Now, if used in this way, these kind of biostatistical indicators of whether a policy works according to one's kind of genetic profile would then also produce knowledge for policy making. So as others have argued, not acknowledging genetically informed explanations for individual differences can lead to suboptimal policy decisions and suboptimal experiences for children in school. So we can understand polygenic scores here then as kind of being wedded with the existing kind of use of extensive numbers and statistics in education policy and governance. Polygenic scores become almost like a kind of governing knowledge that can be used for policy intervention design. The final promise of polygenic scores for policy is their potential to be used as the basis of the prediction of human capital. So we've termed this the geno-econometric biocapitalization, which is more of a mouthful than I thought it was going to be. So here, the interdisciplinary field of genoeconomics is key. So genoeconomics combines economics and genomic sciences to make statistical predictions of the economic value associated basically with different distributions of DNA amongst individuals in the population. So education then is important to genoeconomists because it's taken as a key indicator of later economic outcomes. And this human capital emphasis in educational genomics of the genoeconomic kind is evident in papers like the one you can see here, where it's claimed that educational attainment is a measure of human capital and that governments are seeking to increase levels of educational attainment, to boost basically economic outcomes of various kinds. So genoeconomics researchers, many of which do educational genomics studies, therefore seek to use polygenic scores as kind of predictive econometric instruments to survey, as it were, for biological signals of future human capital in the genome, and then to call for social policy investments based on the results. So what we've called this kind of genoeconometric biocapitalization of children means a forecasting of future economic value from polygenic scores and foregrounding something like future human biocapital as the kind of target of educational investments and interventions. So we come to my fifth eye of educational genomics. Education genomics, as I hope I've indicated, is not only an expanding domain of experimental research. It's also making claims to kind of biological authority in discussions in education, and making normative claims too. But the main point I've been trying to work towards throughout is that educational genomics should be understood as an inventive science. Its underpinning infrastructure and instruments don't merely make the underlying biological substrates of learning outcomes visible, but they fabricate or invent new genomic facts about learning. And they fabricate the idea that learning is basically reducible to molecular interactions amongst thousands of genetic polymorphisms. But, as Marai Kaufman reminds us, DNA does not speak for itself. It never did. Today, digital technologies are more than ever part of making DNA speak. DNA is collected and rendered into digital data by hardware. It is stored in a variety of databases and analyzed by algorithms. These techno-scientific dynamics matter. Together, technology, scientific procedure, and human discretion co-produce how DNA speaks as information and what meaning it will have. Polygenic scores don't speak for themselves either. They are bioinformatic inventions that also invent new biological accounts of learning and of what a student is. Through polygenic scoring, educational genomics has invented a desubstantialized, decoded, statistically calculable student body as a digital object of attention. And educational genomics also potentially reinvents ideas about schooling and education. It poses new normative assertions that decisions about children's schooling should be informed by genetic data, that teachers should take genetic bioinformation into account in their strategies of teaching and differentiation, that education should be about the cultivation of future human biocapital. But these inventions also raise considerable social, political, and ethical challenges. So let me just finish up for the last few minutes with some critical provocations that may invalidate many of the claims of educational genomics. So this is what is referred to at the beginning as a kind of sneaky half-sixth eye of educational genomics, which draws on recent critical accounts of polygenic scoring as a scientifically, politically, and ethically problematic methodology. And the first big issue is non-portability. So the vast majority of genetic bioinformation used in these studies has been gathered from people classified as having white European ancestry. So what is known genetically about learning outcomes or attainment or about intelligence or about cognitive ability is only relevant to a white European ancestry group and is not portable and does not represent other population groups. So if you introduced an intervention into schools based on insights from educational genomics, you would simply have to exclude any other group. So clearly there's a massive issue here of reproducing and exacerbating racialized inequalities that would invalidate many of the proposed interventions just described. And that's very much detailed in a recent Nuffield Council on Bioethics report. The second problem is unknown biology. Educational genomics is entirely a big data exercise. It can only report correlations between genetic variations and educational achievement. So actual mechanistic biological explanations of what's going on inside the body and the brain are really quite absent in educational genomics. There are no causal explanations of how genetic polymorphisms actually affect outcomes. There are only algorithmically calculated correlational claims of their association. And the third problem is invalid claims of genomic salience. So according to critics, social factors remain vastly more explanatory of educational outcomes than genetic predictors. And when I say critics, I mean critics from within the field of genetics itself and within the field of behavior genetics. So the very well-known behavior geneticist Eric Turkheimer, for example, argues that the most recent scientific evidence actually shows that direct genetic effects on educational outcomes are near zero once you fully account for social factors. So after 15 years of research, the genome might not, after all, tell us anything meaningful about the contribution of biology to educational outcomes and learning behaviors at all. So, educational genomics is evidence of the emergence of a kind of a form of bioeducation, bio-edu-data-science as a source of growing authority in education. Bio-edu-data-scientists are now seeking to kind of data mine the body and brain with advanced technologies to sense out the molecular or neuromolecular differences that, it's argued, make a difference in learning behaviors and outcomes. I think it's important that we keep engaging critically with the myriad social, political and technical processes through which they're producing such knowledge claims and their assertions to objective biological knowledge and authority. Otherwise, the genomic testing startups, like our old friend Nucleus, might start staking claims to educational authority and knowledge production too. Educational genomics researchers themselves are already claiming that DNA testing companies are increasingly marketing their products to encourage parents to test their children for educationally relevant traits. So there is now the prospect of such genomic startups becoming part of the knowledge infrastructure of the bio-edu-data-sciences and of making this kind of bio-informationally predictable student the object of policy attention and practice interventions. Thank you very much for your time this morning.
With a big chair. So we have 10 minutes for reactions, questions, clarification. Please.
Thank you very much. I felt like my brain just went to a really good party, so thank you very much for that. I've got a couple of points and I want to comment on it. First, we speak about intelligence, and obviously, as biological markers, we're speaking about IQ, but what about EQ? I mean, we could have rocket scientists, but they could also turn out to be psychopaths. What does the biology and the research say about that? The second thing is you spoke about
Yeah, really, really important points. Yeah, so intelligence here is very much being used in the sense of IQ. So it's taking former kind of psychometric ways of understanding or constructing intelligence and then finding ways to genetically understand them. There's some interesting issues in that people talk about the missing heritability problem. Is the old ways of measuring IQ in behavior genetics often said about 50% of intelligence is heritable. When you run the kind of polygenic score studies that I've indicated here, it drops to about 15%. So it's like, well, where did all their heritability go? You know, so I think it's another indication of some of the methodological problems with this, although there's obviously a longer historical narrative about issues around the construction of IQ. Could polygenic scores be weaponized and used for increasing discrimination? There's an absolutely horrible case of one of the SSGAC's study being included as evidence in a mass shooting incident in the United States a few years ago, as it was claimed evidence of white racial superiority. So very much happening, and organizations like the SSGAC have had to work quite hard to defend some of what they're doing. Daphne Marchenko has written extensively about this, if you're interested. Yeah, is this classifying race and social inequality? I mean, polygenic scores don't really, because they can only account for those who are classified as having white European ancestry. But that's precisely the problem that relates to the horrible incident in Buffalo in the US, is that the lack of genetic predictivity of the polygenic scores for anybody other than white Europeans means it can be used against those from other racialized groups. So yeah, really important points. Thank you very much.
Please.
Sorry, my question is, what about the impact of something like epigenetics? So we know, having poor sleep and a whole lot of behavioural things have big impacts on the DNA, maybe not on the SNPs, but certainly on other features of people. And it seems to me that purely focusing on SNPs is a piece of reductionism that just isn't valid.
Yeah, sure. And, there is internal debate I didn't really have time to go through today about the extent of environmental influence on things and different ways in which that's explained as well. Is it that it comes back into the body? So a lot of these people are looking for the molecular impact of the environment. There's another issue here, I think, which is about the way in which social factors, which I think when we're thinking about education and educational outcomes, are many vast, relational, joined up, and very difficult to apprehend, and how those are often reduced to environmental things that are measurable and observable and so on. So in a lot of these studies they say, well, the gene by environment interactions are really, really important. We mustn't discount the role of the environment, but then the environment gets reduced. It becomes about certain family factors or certain classifiers of a particular kind of sort of neighborhood, or it becomes, or the use of kind of quite crude socioeconomic quantified signals as well. There's not a lot of engagement with ideas of epigenetics specifically in this work, though.
Please. Take a microphone. Thanks.
Thank you very much for the presentation. My question is, we are looking at ways to identify children who adapt well or not well to their city school systems. And my problem is, how could we look at the school system, how should they be adapted at the needs and possibilities of children? So the independent variable is genomics, but it can't be variables aggregate to a very high level. So And maybe it would be more useful if we had more specific information, what kind of competences or what kind of abilities are determined by genetic informations for better be able to adapt to school systems.
Mm. I think there's a difference between the claim that we can collect better biological information and that we could use that to adapt education systems. That's K.P. Harden's argument. And the argument that I and my colleagues are trying to make from a more social scientific perspective, which is what we're actually producing here are bioinformational proxies using methods that don't necessarily correspond with biological reality. Polygenic scores are entirely statistical artifacts. A polygenic score is not in the body. A polygenic score can only emerge from a database. Cecile Janssens, who is a genetic epidemiologist, refers to polygenic scores as creating an entirely new biological reality that doesn't correspond with many other ways in which we understand biology itself. That's why I think there's that important point about as I made towards the end, about unknown biology. That actually as it was explained to me in one of the interviews we did, we understand the genetic architecture of educational outcomes, or educational genomics, people say, the genetic architecture. He said that's basically a statistical artifact. He said we know nothing about the actual biological architecture. We don't really understand what's going on between cells or inside of genes, or how genes really interact. All we know is that these SNPs interact with these SNPs, and together we now have this kind of genetic architecture of educational attainment. So that was the point I was really trying to get at, is this problematic kind of set of assumptions that a bioinformationalized way of understanding a learner or learning outcomes is the same as a biological one, which I think is highly disputed even between sort of geneticists and social or behavioral geneticists.
We have time just for 2 reactions. One colleague here, and you have.
Yes,
please.
thank you. I have a fairly provocative question. So the developments you've outlined here shock us quite a lot, myself very much included. But at the same time, over the last decades, there's been, I think, an increase in the recognition of, for example, neurodiversity. And for example, we might see it as a positive thing that we can medicate students who are struggling with ADHD and so on. So this is something that we, at least to some extent, accept. So my question is, having examined this more maybe future-oriented issue here, do you think there's a slippery socio-technical slope from where we are into these futures, or do you think there's a discontinuity in how much we can reduce these sort of neurodiversity, behavioral biology sort of issues as we study this interface of education and biology, if that question makes sense. Thank you.
Yeah, I'm afraid I don't have a satisfying response to that. The ECER moot, which is happening on Friday at 4 o'clock, I believe, is picking up some of the issues around the what Christian Idison and colleagues call the neuroaffective turn in education. So I think there are some similar issues in neuroscience around, the particular way in which neuroscientific instruments are set up and enacted, and the particular forms of neurodiversity which might become visible or legible and sort of possible to act upon in some ways. And that could, of course, be for, I suppose, some positive reasons. We don't always have to see this in terms of interventions that govern and kind of impose burdens on students. But I think there are some really difficult things to disentangle there. So take it up at the moot on Friday if you want to take it further.
Hi, thanks again for your presentation. It's been very stimulating. There are a couple of points I wanted to make. The first one is I didn't quite get how they set up the correlation between genetic information and educational attainment, because it depends on when that information gets collected. It doesn't take into account socialization, differences in educational experience. So that's the first point. The second one is when you made the point about that the bioeducational data is based on white people, it also does not take into account, or it assumes that white people are homogeneous and there are no differences between them. And then the third question that I have for you is, while I get the sense that there's an attempt to set up social inequalities or the consequences of social inequality, what about the taking into account the biology, which says that we are all alike, we all come with the same information, the same kind of genetic base, and that's the basis on which there's actually equality, and what makes the difference is socialization.
Okay, yeah, great points. I'm just going to pick up on one of them, I think. And that's the issue around, yeah, there is, of course, diversity in what are classified as white European ancestry groups. And the construction of ancestry groups in genetics is itself a deeply problematic exercise. I think what I would say in relation to these studies specifically is, I said the SSGAC is the reference for many other studies because it's collected the massive data sets, and it collected them from the UK Biobank, and it collected the data from 23andMe. So that's a very, very specific kind of population. That's basically white middle-class people from the UK who have chosen to donate some of their information to a publicly funded science project in the UK. And when we think about 23andMe, that means we've got basically a Silicon Valley population. It's white, educated, wealthier than average, willing to give up data. So there are distinct issues with the kinds of data that are included in the massive data sets which are being used as a reference against which then kind of local cohort data are assessed. So if you're trying to work out a polygenic score for thousands of school children somewhere in the East Midlands in the UK, you can spit swab them and so on. What you then have to do is you have to run those data against the established polygenic scores that have been produced by organizations like the SSGAC. So there's a kind of cascade of issues here in terms of samples, the way the polygenic scores are produced, and then how other studies are built upon them.
This is a very challenging issue, but we have a time just for one more reaction. Please.
Okay, thank you. You've pointed to the difficulty of genetics kind of representing the actual person, and that that's never possible. What I'd like to ask is, do you think that's just not a continuation of all attempts to assess people, to kind of make representations of people? Isn't that what we've always done in education? We have testing and assessments, and we make claims that this tells you who this person is and what we need to therefore do. And so my question is whether there's anything sort of unique in that sense, you think, about this sort of genetic attempt to do it, as opposed to all the other things we've done historically.
I think the significance here, to some degree, is the claims that we're getting at something that is biologically real, that goes beyond traditional kind of psychometric constructs or the data that we get from taking observations. That is certainly the claim that is made by many of the people who are working in this kind of space. We've always had to rely on constructs before. We've also always had to rely on things like observations of behaviors, as in kind of conventional behavior genetics. And the argument made by many people working in this space is, I think one of the quotes I read out said, we are now developing a more biologically realistic understanding of school achievements, intelligence, cognitive abilities. There's a huge claim here. I'm trying to some degree, kind of deconstruct that claim and say, actually, these claims rely on a kind of socio-technical cascade of technologies, instruments, the way those instruments are tuned, the kinds of methods that those instruments allow these scientists to enact and to undertake. And all of those actually shape a particular kind of configuration of what we might understand learning to be. So I'm trying to take that claim of biological realism to task, though at the same time recognizing that all forms of data that we collect in school also construct the student in a particular kind of way.
Thank you, Ben. Thank you all. And at the end, a small gift for the Ben from the ECER for his contribution.
Thank you. Thank you very much.
He's a Nobel Prize author.
Okay, thank you everyone.